Good morning everyone, we are Group 2 and we will discuss to you about the real concept of genes, transmission, recombination, mutation, and function. For the introduction, the gene has a long and storied history. Understanding the current use and misuse of the term gene necessitates an understanding of how it came to be in the vocabulary in the first place by Bartol 2011, in 1865 by Gayon 2016. Gregor Mendel, a Moravian researcher and Augustinian monk, established the frameworks of present-day hereditary qualities with his milestone investigations of heredity in the nursery P, or Pisum sativum. Mendel did not want to offer general laws of heredity, but only a law of the development of hybrids in plants. Furthermore, Mendel's memoir remained largely unknown until 1900s, when his laws were rediscovered by Gayon 2016. Through the rediscovery of Mendel's law, it led the other scientists to investigate deeper and discover the genes. A gene is a short section of DNA that serves as the basic physical and functional unit of heredity. Some genes function as blueprints for protein synthesis. Many genes, on the other hand, do not code for proteins. Through the years of experiment discovering the concept of heredity and with the advancement of technology, scientists can understand the gene concept and correct the misconception concerning the concept of genes taught by scientists in the past. In line with that, this study aimed to identify the development of the concept of genes, to fully understand the gene concept and eliminate the misconception attached to it through a systematic review, a review of published literature related to the chosen topic. For the objectives of the study, generally, this review aimed to understand the development of the concept of genes. Specifically, the review sought to achieve the following. First, briefly describe the origin of genetics. Second, discuss the following definition of genes throughout history. A. Gene as a discrete unit of heredity. B. Gene as a distinct locus. C. Gene as a blueprint for a protein. D. Gene as a transcript code. And lastly, explain the gene as a unit in terms of transmission, recombination, mutation, and function.
For the materials and method, the search and selection process of the study has used a scientific electronic search that was conducted in Google Scholar and Semantic Scholar that serve as the search engine. Published articles and books related to our topic from 1930 to 2021 were used in this study. There are 7 papers that were chosen from a total of 31 articles found through a search engine and cited references from various publications. The 7 articles was analyzed with the use of the following inclusion criteria. Number 1, it had to be written or translated in English. Number 2, it had to be published between 1930 to 2021. Number 3, any studies concerning the stated objectives. And number 4, it had to be available in full text. This use of parameters in the selection of literature to be reviewed is adapted from the study of Padel et al. 2020 and Rituya 2020. The data collection method for the study was a scientific review. The purpose of the literature review is to collect and analyze all information about the issue from published articles. This study conducted a literature review on the concept of genes. The downloaded articles were then renamed using the code number and saved in a folder. The screening was performed by researchers using inclusion criteria that was mentioned earlier, and articles that met the inclusion criteria were stored in separate folders. The articles that did not meet the inclusion criteria were then deleted. The researchers then reviewed the logs after verification and listed the data of the gene concept pertaining to transmission, recombination, mutation, and function. The data was then analyzed and interpreted in the next section of this study.
For the results and discussion, these are the 7 individual profiles of reviewed published articles and books related to the development of gene concepts. Jean Gayon and his article From Mendel to Epigenetics, History of Genetics in the year 2016, Peter Portin and his article Historical Development of the Concept of the Gene in the year 2002, H.P. Gurushankara and his article Genes, Concepts and Applications in the year 2015, Peter Portin, The Concept of the Gene, Short History and Present Status in the year 1993. Peter Portin, in his article The Origin, Development and Present Status of the Concept of the Gene, a short historical account of the discoveries in the year 2002. Peter Portin and Adam Wilkins in their article The Evolving Definition of the Term Gene in the year 2017. And lastly, Gerstein et al. In his article What is a Gene? Post-ENCODE History and Updated Definition in the year 2007. Table 1 shows the profile of each of the study's 7 peer-reviewed journal articles and books. Each article meets the inclusion criteria of having been published between 1930 to 2021, being in full text, written in English or translated into English, and containing knowledge relevant to the study's goals. Origin of genetics, a brief history. The development of genes started from the establishment of genetics that brought the attention of many geneticists to dig deeper into the study of heredity. Gregor Mendel established the frameworks of the present-day hereditary qualities with his milestone investigations of heredity in the nursery P. Pisum sativum, by Mendel 1866 as cited in Portin and Wilkins 2017. Meanwhile, the term genetics was coined by the English biologist William Bateson to refer to the science of heredity and variation. He proposed naming the chair created for him in Cambridge in 1906 after the word genetics, but it was rejected, and eventually, Bateson's chair was named Chair of Biology. But at the Third International Conference on Plant Hybridization, he proposed that the new science of heredity based on Mendel's laws be called genetics, and this was enthusiastically accepted. The intention of Mendel was not to create a general law of heredity but only laws on the development of hybrids in plants. Other scientists did not value it at the time because they did not understand Mendel's concept, but it was not until 1900s that Correns, Chermak, and Revised 1900s independently observed the same rule of segregation and independent assortment that Mendel had discovered 35 years earlier. And Correns proposed the name Mendel's law for these rules of inheritance, 2002. In the present day, Mendel's law was composed of 3 laws, namely law of segregation, law of independent assortments, and law of dominance. The first law is the law of segregation. 2 alleles of a gene separate or segregate and enter gametes separately, with half of the gametes carrying one allele and the other half carrying the other allele or haploid. 2 gametes fuse to produce a zygote with 2 alleles or diploid. The second law is the law of independent assortment. Due to independent assortment during meiosis, alleles of genes that govern 2 different characters segregate independently during the formation of gametes. The inheritance pattern was derived from the ratios 3:1, 9, 3, 3, 1, 1, 1, 1, 1. As a result, genes were considered a statistical entity. Lastly, the third law is the law of dominance. One allele in a heterozygote may conceal the presence of another. This principle is a statement about the function of human genome. Even when there present single copy, some alleles control the phenotype.
The next part of the results are defining the history of genes. In 1860s to 1900, gene is described as a discrete unit of heredity. Wilhelm Johansen coined the term gene in 1909 that was derived from the term pangene, a term coined by Hugo de Vares to describe things involved in Darwin's hypothetical mechanism of inheritance, which is pangenesis. In Mendel's study, he demonstrated that the variation of traits passed on by offspring to the progeny were caused by the variation in inheritable factors. He regarded it as factors that one initiate the variation of traits inherited. The works of Mendel were not given attention until 1900 and were rediscovered by 3 botanists, Karl Correns of Germany, Hugo de Vares of Netherlands, and Erich von Chermark of Austria. They all independently cited and discussed Mendel's experiment as fundamental to understanding the nature of heredity. It was then concluded that the genes is the unit of heredity, but Johansen viewed the gene as a calculating factor only. He avoided a speculation on its physical characteristics and wished this unit of heredity to be free of any hypothesis regarding its physical or chemical nature. In 1910s, gene is described as a distinct locus. In 1910, Morgan and his student Bridges, Miller, and Sturtevant were looking at how mutations in Drosophila melanogaster segregated, and under findings using a model, they were able to explain that genes are arranged linearly and crossover ability is proportional to the distance between them. In 1913, Sturtevant created the first genetic map by mapping 6 Drosophila melanogaster sex-linked genes into linear order. That draws attention to the fact that the linkage group linear structure matched that of the chromosome. However, it is only circumstantial evidence in support of the chromosome theory of inheritance. Bridges obtained the first direct evidence in 1916 when he was able to show that a specific abnormal behavior of sex-linked genes or genes that reside on the sex chromosome and Drosophila melanogaster, namely non-disjunction, corresponded to the analogous non-disjunction of the sex chromosomes. According to the findings of Bridges, 1916, it was then proved that genes reside in the chromosomes, making genes known to be located inside the chromosome. Morgan, together with his students, using the findings of their experiments, created the chromosome theory of inheritance, according to which the genes reside in the chromosome like beads on a string. In 1940s, gene is described as a blueprint for a protein. According to the study of Bedell and Tatum in 1941, mutation in genes can cause defects in metabolic pathway. This was originally referred to as the one gene one enzyme viewpoint, which was later changed to one gene one polypeptide. Bedell and Tatum in 1941 and SRB and Horowitz in 1944 significantly elucidated the nature of gene function when they demonstrated using Neurospora crassa as their experimental organism that genes control enzyme synthesis and that each gene is responsible for the synthesis of a single enzyme. According to this theory, each gene regulates one synthesis or activity of a single catalytically active protein. Based on the remarkable association found in Neurospora between mutations of specific genes and the blocking of specific biosynthetic steps. Bedell in 1945 assumed that every biochemical reaction had a specific gene controlling its course and that the gene's primary and possibly sole function was to direct the configuration of protein molecules. That was then the start of treating genes as the blueprint of protein, because it was before thought that genes produces proteins. In 1950s, gene is described that has a physical molecule. In 1952, Hershey and Chase showed that DNA was responsible for bacteriophage multiplication. In their study of the protein and DNA of virus T2, they concluded that the DNA evidently entered the bacterium and played a part in virus multiplication. In 1955, Hershey and Chase established that the substance transmitted by bacteriophages to their progeny is DNA and not protein. Through the study of Hershey and Chase, it was clear that the genetic material of viruses is its DNA, and so it was suggested that it is the same on the higher form of life. A gene is a little piece of DNA, and so DNA, the physical molecule of a gene, was responsible in transmitting the genetic material of an organism. In 1960s, gene is described that has transcribed code. Watson and Crick solved the three-dimensional structure of DNA in 1953, which explained how DNA could function as a heredity molecule. But earlier in 1948, Bovian and colleagues conducted chemical analysis of the DNA content of the nuclei of the domestic cows, thymus, liver, pancreas, and kidney. As a result, two remarkable discoveries were made. First, the DNA content of the nuclei of the various tissues were discovered to be essentially the same. Second, the DNA content of sperm nuclei was discovered to be roughly half that of diploid tissue nuclei. The discovery that the DNA content of nuclei is unaffected by tissue or individual and is solely dependent on chromosome content, is a strong argument in favor of the theory that DNA is the carrier of heredity factors. Chargaff discovered an important rule governing the proportions of DNA bases known as Chargaff rule. It was discovered that the DNA invariably contained equal amounts of adenine and thymine on one hand and guanine and cytosine on the other, which suggested that the base pairing rule is the structure of DNA to Watson and Crick. The double helix model of DNA was proposed by Watson and Crick in 1953. DNA met all the requirements for genetic material including auto-replication, specificity, and information flexibility. The linear structure of DNA determines its specificity and information content. The sequence of nucleotides in DNA constitute genetic information that is coming from the genes. The genetic code suggested by George Gamow is made up of 3 nucleotides per amino acids. Because there are 20 amino acids and only 4 bases, he reasoned that the coding units could not be single, or 4 combinations, or pairs only 16 combinations. Rather, he believed that triplets or 64 possible combinations were the genetic code's coding unit. However, he claimed that the triplets were non-degenerate and overlapping. Marshall, Narenberg, Severino, Okowa, Cobbett, Corona, and their collaborations deciphered the genetic code, confirming the collinearity hypothesis biochemically.
Gene as a unit in terms of 4 genetics phenomena are used to operationalize the gene: genetic transmission, genetic recombination, gene mutation, and gene function. The gene was widely regarded in the classical view as an invisible unit of genetic transmission, recombination, mutation, and function, with all of these criteria leading to the same unit of genetic material. Gene as a unit in terms of transmission. According to Pertin 1993, Mendel 1866, and the 3 rediscoverers of his principle, Correns 1900, De Vries 1900, and von Chermak 1900, inferred and clearly understood the gene as the unit of genetic transmission, the transfer of genetic information from one generation to the next, in what is now known as Mendel's first law or the rule of segregation. When a hybrid AA produces gametes, each gamete contains either A or A, but never AA or no A factor at all, according to this rule. In other words, during the formation of gametes, the alleles of the gene segregate, which means that each gamete includes 1 unit of each gene. Gene as a unit in terms of recombination. In defining recombination as a unit of the genes, it is important to distinguish interchromosomal and intrachromosomal recombination by Pertin 1993. The interchromosomal recombination holds Mendel's second law or the rule of independent assortment. This rule states that when an AABB x AABB cross-produces gametes, the hereditary factors A and B are distributed equally among the gametes in the combination in which they are entered the hybrid. In S, the parental combination of AB and AB are equally as common as the new combinations AB and AB. In intrachromosomal recombination, genes that are so far apart on the chromosome that are chiasma, the point of contact, the physical link between 2 chromatids belonging to the homologous chromosomes, always forms between them follow the rule of independent assortment, while genes that are closer to one another follow the rule of linkage. In parental combinations, the genes are distributed among the gametes more frequently than in recombinations, which is known as linkage. Morgan's argument was taken a step further by Sturtevant 1913. Crossing over was thought to happen between genes but not within them at first, so recombination became a defining criterion for a gene. Binzer called the smallest unit of recombination the recon. It is the smallest unit of genetic materials that can be separated from other such units through genetic recombination, but which cannot be divided further, meaning it will not undergo mitosis or meiosis.
For mutation, the fact that mutant alleles of a single gene were mutually selected revealed the gene as the unit of mutation. As a result, the gene was thought to change as a single unit in the event of mutation. Viverys coined the term mutation in 1901 to explain a speciation by sudden appearance. According to Porty 1993, Hermann Joseph Müller is responsible for the modern theory of mutation and its implication for the theory of the gene. Müller's ideas are most clearly expressed in his paper from 1922 and 1923, in which he raised several points about the nature of the gene, as well as the concept of mutation as a change in the gene. Drosophila melanogaster is one of the most important insects in the world, and Müller's 1927 and 1928 observations that Xers greatly increased the rate of mutation in these insects was strongly supported by his data. He proposed that a mutation usually affects only one specific gene at a time. The linear relationship between dose and induced mutation rate suggested that the radiation quanta or released electrons were acting directly on the gene thought to be the target of radiation. As the effect of radiation were quantified, genes became real units of genetic material. In reality, the situation was more complicated because the target theory proved insufficient later on, and there were also indirect effects of radiation on genes. Huynot made a significant contribution to the theory of the gene, particularly the theory of the gene as a functional unit, in 1904. While studying the inheritance of coat color in mice, he discovered that the number of Mendelian alternatives for a given character was not limited to 2. As a result, he discovered the phenomenon of multiple allelism. Benzer called the smallest unit of mutation, or the muton. It is the smallest unit of genetic material in which a change is sufficient to cause a mutant phenotype. And lastly, perfunction. One of the first attempts to represent an organism as a composite of particulate hereditary determinants was Mendel's recognition that the germinal element transmitted the differentiating characters of his pea plants. The gene, as the unit of function, was not indivisible. Within the gene, independently mutating mutation sites existed, which could be separated from one another by utilizing genetic recombination. When George Bedell and Edward Tatum 1941 and Adrian SRB and Norman Horowitz in 1944 demonstrated using Neurospora crassa as their experimental organism, they substantially defined the nature of gene function, that genes control the synthesis of enzymes and in particular that each gene is responsible for the synthesis of one single enzyme. Benzel coined a new slang term, cistron, named for the genetic function unit. With the help of the cis-trans test, the cistron is operationally defined. The cis-trans test compares cis and trans heterozygote, and mutations in the cis heterozygote are on the same chromosomes, whereas the trans heterozygote was homologous chromosomes. As a result, the cis genotype heterozygous is as like this, while the trans genotype heterozygote is as like this. If the cis heterozygote has a wild-type phenotype and the trans heterozygote has a mutant phenotype, A and B are cis-trans mutation. If both cis and trans heterozygote have the same phenotype, then the B are mutations in different cis-trans. The cis-trans is a synonym of the gene, but this term should be used only when it is based on cis-trans tests or biochemical evidence, which will be dealt with later.